This section is on eukaryotic cells, and they're much more complex than the prokaryotic cells we talked about in the last lecture. So this is going to go kind of fast. Remember that you can watch this video again and pause it and get more information and slow it down a little bit for your pace. But we have a lot of organelles to cover, so we're going to cover them pretty quickly. Eukaryotic cells, they're complex. They have a nucleus inside of them, and they have membrane-bound organelles, unlike the prokaryotes. Animals, plants, fungus, and protists are all examples of organisms that are made up of eukaryotic cells. Okay, the cell walls. Some of the eukaryotic cells have cell walls. Plant cells usually have a primary and a secondary. Cellulose is what makes up the primary, and we can find these in algae, which are protists as well. Lignin is found in secondary cell walls. Fungi are a little bit different. Some of them have cellulose, and some of them also have a substance called chitin, and chitin is found in the exoskeletons of insects as well. Okay, so this is eukaryotic structures in animal cells and plant cells, and this is going to help you in a couple of your assignments. Notice that we talk about the cell walls, what they're made up of, the function, and it goes through and tells you on each one of these organelles. There's another one a little bit more detailed. This is a picture, a generalized picture of an animal cell. And you're going to color some pictures. You're going to have to identify some of the organelles in the animal cells. And you can find this picture in your textbook as well, with all of these already labeled. If you notice the ones that have an asterisk beside them, the lysosomes, the centrioles, they are not found in plant cells, but they are found in animal cells. This is a typical plant cell. And again, you're going to become familiar with these organelles and the ones that are in plant cells and in animal cells as we go along throughout the chapter. Notice the asterisks here are not found in animal cells. So cell walls, ingranum, chloroplast, and this huge central vacuole are not found in animal cells. Let's take a look at each one, the organelles in a little bit more detail. The function of the nucleus is to store DNA, and all eukaryotes have a nucleus. The DNA is stored in a form called chromatin, which is long and strung out, and this is what makes proteins. This is also, the chromatin is what divides to make chromosomes. They coil and supercoil to make chromosomes. There's a nucleolus, at least one, usually inside of every nucleus, and this is where RNA is made. Then it's surrounded with a nuclear envelope, and the nuclear envelope has pores in it. So if you can kind of imagine a wiffle ball around the outside, and there's a purpose in having these large holes in the outside of the nuclear envelope, and that's due to these large molecules that are going to come out of the nucleus that are made by the nucleolus. So we have the nuclear envelope around the outside with the pores in it. We have chromatin and the nucleoplasm on the inside. The nucleoplasm is similar to the cytoplasm, but it's just found in the nucleus. And we have at least one nucleolus that makes the RNA. Notice that this double-layered membrane, the envelope, is double-layered just like the cell membrane is. Ribosomes, if you remember, are found in both prokaryotic cells and eukaryotic cells. Their function is to be the site of making proteins or protein synthesis. The messenger RNA is used as a template for that, and there are two sections to the ribosome, a small subunit and a large subunit. Those subunits are made up of rRNA, which is ribosomal RNA, and protein molecules. They are found attached to the endoplasmic reticulum, or the ER, and they also can be found floating throughout the cytoplasm. Okay, the endomembrane system. This consists of the nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, and some vesicles, which are just tiny membranous sacs. The function is that they are used as transportation and product processing sections of the cell. And this also helps us to compartmentalize the cell, similar to the way your rooms in your house compartmentalize your entire house instead of everything just being open. This is a picture of the endoplasmic reticulum. It's considered the ER, is another abbreviation for it, but you need to know it is the endoplasmic reticulum. And if you notice, this is a micrograph of it, and it shows you what it looks like. But if you notice here, we have ER that the purple runs right into the red one. The red one has no ribosomes, so it's considered smooth. The purple color has ribosomes, and it's considered rough. So a little bit more into the endoplasmic reticulum. On the rough ER, it has ribosomes, and its job is to process and modify proteins. On the smooth ER, there are no ribosomes at all, and this is where phospholipids are made or synthesized. And there's a lot of other functions for the smooth ER as well. One of the things I want to mention about the ER, let's back up a couple of pictures here. This actually attaches to the nuclear envelope on one end, and it will attach to the cell membrane on the other end. So it's kind of a passageway as well. The Golgi apparatus is the next organelle. It is referred to as the Golgi apparatus, the Golgi body, and the Golgi complex. So any of those names are acceptable. It's considered the post office of the cell because its job is to package proteins and lipids. The proteins are made in the rough ER, that's RER, so rough ER. They have tags that we consider zip codes, and they direct the Golgi apparatus to where to send them, whether it would be someplace inside the cell or for export outside the cell. So this looks like the Golgi apparatus here. So we have lipids in this case that were produced, and they're going to be packaged by the Golgi body, and they are ready for export outside the plasma membrane. The lysosomes are the next organelle. These are made by the Golgi apparatus, and they have enzymes inside of them. And your book considers them garbage disposals of the cell. I consider them the cell eaters because their job is to break down dead, unwanted parts of the cell or foreign invaders that come inside of the cell. And if the cell were to die, the lysosome would open up and the enzymes inside of it would digest the chemicals that are within the cell. Okay, let's look at the vacuole. The vacuoles are large membranous sacs, and the larger the vacuole inside depends on the plant cell. Now we have some smaller vacuoles inside of the animal cell, but the one inside the plant cell is rather large because we need photosynthesis to occur. The job is to store substances such as water, pigments, and toxins. Peroxisomes are membrane-bound vesicles, and they have enzymes inside of them similar to a lysosome. They break down molecules producing hydrogen peroxide, and they immediately get them broken down into water and oxygen. And if you notice, it's not really very significant looking inside of the cell here. This is more what it looks like in a micrograph. We discussed in the last chapter that life is possible only because of a constant input of energy. Chloroplast and mitochondria are energy-producing organelles. There are mitochondria inside of plant cells as well as animal cells, but chloroplasts are only found in mainly in plant cells. So solar energy is converted through the chloroplast into carbohydrates, makes sugar. That is taken into the mitochondria and makes ATP. ATP is the energy currency for our cells. So this is the reaction of photosynthesis, solar energy, carbon dioxide and water, and you make carbohydrates and oxygen. On the other hand, cellular respiration happens in all organisms, not just in plants, algae, and cyanobacteria, and it looks like the reversal of photosynthesis, but it's not exactly the reversal. The carbohydrates and oxygen are put together to form carbon dioxide, water, and gives off energy. The big difference here is that the input is solar energy, the output is energy in the form of ATP. Let's take a deeper look at chloroplast. This is where photosynthesis happens inside of plants and algae. And chloroplasts are double-membraned organelles. The stuff inside of it that's kind of like the cytoplasm of the chloroplast is called stroma. They have stacks of thylakoids called grana, and the chlorophyll are actually located inside of the thylakoid membranes. So photosynthesis happens inside the thylakoid membranes. They do have a single circular DNA molecule inside the chloroplast. That may, some of its own proteins are made there, but not all of them. It just depends. The nucleus can make some of those proteins. It does contain some ribosomes. And the evidence of this information, with it having a little bit of DNA and also having some ribosomes, leads us to believe that at some point the chloroplast was a single organism, a prokaryotic type of organism, got incorporated into another cell and became an organelle instead of just an individual prokaryotic cell. All right, so this is a chloroplast inside the cell. This is what it would look like in a micrograph. And if we look here, these stacks right here are called grana. These are, each one is a thylakoid, and the gel-like substance in between is the stroma. Mitochondria are found inside of all eukaryotic cells, plants and animals as well. They are the site of cellular respiration, and they have a double membrane. They have a matrix, which is similar to the cytoplasm. The folds are called the cristae, and this increases the surface area so more ATP can be produced. And again, just like the chloroplast, they have their own DNA, which leads us to believe that at some point the mitochondria actually may have been a prokaryotic cell and was incorporated into another cell and has a specific function. This is what a mitochondria looks like, and these folds are the cristae that increases the surface area for ATP production. The matrix is the gel-like substance inside of it. The job of the cytoskeleton is to keep the shape of the cell, and we used to think back in the day that that was just a gel-like substance, and now we know differently with the advancement of technology and being able to see smaller and smaller things. We realize it's actually made up of some protein components that are actin, filaments, intermediate filaments, and microtubules. The actin filaments. There are actually two chains, and it's twisted very similar to DNA. They are found in your intestinal villi. They help make pseudopods, which is a false foot, just a single cell that can actually reach out and it looks like a foot, but it's just movement of the actin molecules. It's also found inside of muscle cells. Actin and myosin work together to form muscle contractions. So it interacts with myosin molecules in muscle cells, and you have to have ATP in order for this to happen, and it causes contraction. Intermediate filaments, there's a little bit of a size difference. Their function is to support the nuclear envelope and cell-to-cell junctions that hold your skin tightly together. And it also strengthens human hair. The microtubules are hollow cylinders of tubulin, alpha and beta tubulin. They are under control of microtubule organizing center, which is the MTOC. And the most important is the centrosome, and we'll talk about the centrosome as we go throughout the cell. And there's all three of those together. This interacts with motor molecules. It's called kinesin and diin, and it causes the organelles to actually have movement. The centrioles are located inside of animal cells, and I always thought they kind of looked like a churro. And there's a couple of them inside the animal cells. And they have a 9+0 tubular pattern. Cilia and flagella are projections, and their function is to aid in cell movement. These are projections of the cell membrane. In eukaryotic cells, the cilia are shorter than the flagella. Both of them are membrane-bound cylinders in a 9+2 pattern of microtubules, and this shows examples of that. That's all of the notes that your book provides for you on organelles. So this PowerPoint and this presentation goes along with your textbook notes, so you can fill those in if that's the way that you learn best. I'm also going to be putting a Bozeman cell video into the folder for advanced biology that's going to be very helpful and a lot more entertaining than what this is. So you probably should watch that. And we're going to be practicing learning what the organelles are, what they do, what they look like, where they're located, and how they work together to form the life function of the cell.